Hello, everyone. My name is Haley Bearden, and I'm from Baylor University. I will be talking to you about the discovery and identification of the Lakshmi Bacteriophage from Plano, Texas. A little background. Phage hunting in the sea has broadened to cover 13 to no bacterial hosts, including Arthrobacter. This has allowed a variety of genomes to be compared over time. So the methods that we use to find Lakshmi Arthrobacter phage are isolation and characterization and annotation. First, let's start with isolation and characterization. We began by isolating this bacteriophage from soil and with a direct isolation. Then we enriched the sample and purified it. As you can see in the results section on the first picture to the right is an image of the plaque morphology, which are clear lytic plaques. And then following after amplification and DNA extraction, you can see in the center of the screen the PCR using the 3 tape measure protein cluster specific primer mixes. And that with this, we identified our bacteriophage Lakshmi as an AK cluster. And this was also verified by sequencing. In lane 1, you have AO, AR, AS, and AT clusters. In lane 2, we have AN, AP, AU, and AV clusters. In lane 3, we have AK, AL, AM, and AQ clusters, which were all controls. And then we had the same clusters in 4, 5, and 6, respectively, along with Lakshmi DNA, with 4 and 5 coming up negative, and lane 6 at around 590 base pairs, where you can see a clear band being where AK is. So that helped us determine that our phage was a AK phage. So furthermore, we did characterization of our phage. And also you can see up to the right on the screen under results that an image of our bacteriophage with the TEM, classifying it as a Cyphoviridae phage with a capsid of 50 nanometers and a tail of 100 nanometers. Furthermore, we did annotation of this genome. We were able to identify the genome with a 44,018 base pairs with a G to C content of 60.8% and 60 potential genes. We were able to find the start and stop, and we were able to assign function for some of the genes. As you can see in the Famerator map that has 3 different sections on your screen, the first section is the gene 3 deletion, because originally it was classified as a reverse gene. However, we decided to delete the gene instead of flipping it to be a forward gene because of the ability to extend gene 4. Then you can see in the second image of the Famerator that there is a confirmed conserved frameshift with genes 16 and 17. Additionally, you can see in the third image the potential frameshift of gene 47. To continue, we were able to, along with Famerator, use other programs with annotation such as Pecan, NCBI, PhagesDB, DNA Master, HHPred, StarterRater, Glimmer, and Gmark. This also helped assign functions, which you can see broken down in the chart below, with 51.7% no-known function, replication at 20%, structure at 13.3%, capsid assembly at 6.7%, tail assembly at 5%, and miscellaneous at 3.3%. We had 31 genes with no known function, and 29 genes were found to code for proteins with predicted functions, such as endolysin, resolvase, recombinase. We had 81 total members of the AK cluster now, including Lakshmi. So, what are some future directions that we can go? We can ask, is the potential frameshift around gene 45 conserved, and how does the frameshift affect the function of the gene? Also, we can ask, what are the functions of the genes currently with no known function?